Good afternoon, class. Unit 13, Chapter 21, Part B, the immune system, innate and adaptive body defenses. Okay, so now we're going to look at Part 2, the adaptive defenses. Last class we looked at the innate defenses. So the adaptive immune system is a specific defensive system that eliminates almost any pathogen or abnormal cell in the body. Its activities are that it amplifies the inflammatory response and activates complement. There's a shortcoming which must be primed by the initial exposure to a specific foreign substance. So the priming takes time. Characteristics of adaptive immunity is its specificity, where it recognizes and targets specific antigens. It is systemic, and it's not restricted to the initial site that's been attacked. And it also has a memory component, which mounts an even stronger attack to known antigens upon a second and subsequent exposure. There are two main branches of the adaptive system. One is the humoral immunity, which includes the production of antibodies, and the second one is cellular or cell-mediated immunity. Humoral immunity, like I said, involves the production of antibodies, which are produced by lymphocytes, and they circulate freely in body fluids. They bind temporarily to the target cell, temporarily inactivate the target cell, and mark it for destruction by phagocytes or complement. Humoral immunity has extracellular targets. The second component is cellular immunity. Lymphocytes act against the target cell directly by killing the infected cell, or indirectly by releasing chemicals that enhance the inflammatory response, or activating other lymphocytes or macrophages. Cellular immunity has cellular targets. So let's start talking about, begin the discussion by talking about antigens, and then we'll talk about the antibodies. So section 21.3 is antigens. What is an antigen? It's any substance that can mobilize adaptive defenses and provoke an immune response. It targets of all adaptive immune responses. Most are large, complex molecules not normally found in the body, so they are seen as non-self. Characteristics of antigens, they can be a complete antigen or an Hapten, which is incomplete. And they contain antigenic determinants, and they can be a self-antigen. Antigens can be complete or incomplete. So a complete antigen has two important functional properties. One is immunogenicity, which is the ability to stimulate the proliferation of a specific lymphocyte. And the second one is reactivity, which is the ability to react with the activated lymphocytes and antibodies released by immunogenic reactions. Examples of complete antigens are foreign proteins, polysaccharides, lipids, and nucleic acids. And they're seen on many foreign invaders such as pollen and microorganisms, things like viruses and bacteria. Incomplete antigens, also called Haptens, involve molecules too small to be seen, so are not immunogenic by themselves. Examples would be small peptides, nucleotides, and some hormones. They may become immunogenic if the Hapten attaches to the body's own proteins, and this combination of protein and Hapten is then seen as foreign. And this causes the immune system to mount an attack that is harmful to the person because it attacks the self proteins as well as the Hapten. Good examples of Haptens are poison ivy, animal dander, detergents, and cosmetics. Antigenic determinants are parts of an antigen that antibodies or lymphocyte receptors bind to. The most naturally occurring antigens have numerous antigenic determinants that mobilize several different lymphocyte populations and form different kinds of antibodies against them. Large chemically simple molecules such as plastics have little or no immunogenicity. Figure 21.7, most antigens have several different antigenic determinants. So here we see an antigen in red, and notice that it has several antigenic determinants to which different antibodies will bind to. Self-antigens and the MHC proteins. So self-antigens, all cells in our body are covered with a variety of proteins that are located on the surface that are not antigenic to self. In other words, our immune system will not attack these proteins, but may be antigenic to others in transfusion reactions or in grafts. One set of important self-proteins are a group of glycoproteins called the MHC proteins, and they're coded by genes. They're called the major histocompatibility complex, the MHC, and they are unique to each individual. They contain a groove that can hold a piece of the self-antigen or foreign antigen, and the T lymphocytes can recognize only antigens that are presented on MHC proteins. 1.4 Lymphocytes and antigen-presenting cells. The adaptive immune system involves three crucial types of cells. Two types of lymphocytes called the B lymphocytes or B cells that are responsible for humoral immunity, and the T lymphocytes or T cells that are responsible for cellular immunity, and antigen-presenting cells that do not respond to specific antigens but play an essential auxiliary role in immunity. Lymphocytes. So lymphocyte development, maturation, and activation. T and B lymphocytes share common development and steps in their life cycles. 5 general steps are: 1. Origin. Both of these lymphocytes originate in the red bone marrow. 1.8 Lymphocyte development, maturation, and activation. So number 1 is origin. Both B and T lymphocytes, the precursors of these lymphocytes, originate in the red bone marrow. Maturation. So lymphocytes are educated in a 2 to 3 day process and mature in primary lymphoid organs, so the B cells in the bone marrow and thymus. Lymphocytes are educated for 2 reasons. 1, for immunocompetence. Lymphocytes must be able to recognize only one specific antigen. B or T cells display only one unique type of antigen receptor on the surface when mature, so they bind only to one specific antigen. And second reason is for self-tolerance. Lymphocytes must be unresponsive to their own antigens. So again, figure 21.8, if we look at maturation, so lymphocyte precursors destined to become T cells migrate in the blood to the thymus and mature there. B cells mature in the bone marrow, and during maturation, lymphocytes develop immunocompetence and self-tolerance. Seeding secondary lymphoid organs and circulation. Immunocompetent B and T cells not yet exposed to antigen are called naive. Exported from primary lymphoid organs, bone marrow and thymus, to seed or colonize secondary lymphoid organs like the lymph nodes, spleen, etc. This increases the chance of encounter with an antigen. 1.8 Seeding secondary lymphoid organs and circulation. So immunocompetent but still naive lymphocytes leave the thymus and bone marrow. They seed the secondary lymphoid organs and circulate through blood and lymph. 4th, antigen encounter and activation. So naive lymphocytes' first encounter with an antigen triggers the lymphocyte to develop further. Lymphocyte is selected to differentiate into active cell by binding to its specific antigen, and this is referred to as clonal selection. If the correct signals are present, the lymphocyte will complete its differentiation into the active cell. So if we look at this figure, we have antigen encounter and activation. So when a lymphocyte's antigen receptors bind its antigen, that lymphocyte can be activated. Proliferation and differentiation. So once selected and activated, the lymphocyte proliferates. It forms an army of exact copies of itself, and these are referred to as clones. Most clones become effector cells that fight infections. A few will remain as memory cells, so they're able to respond to the same antigen more quickly the second time it is encountered. The B and T memory cells and effector T cells circulate continuously. So again, figure 21.8, Step 5, proliferation and differentiation. So the activated lymphocytes proliferate, multiply, and then differentiate into effector cells and memory cells. The memory cells and effector T cells circulate continuously in the blood and in lymph and throughout the secondary lymphoid organs. Antigen receptor diversity. So genes, not antigens, determine which foreign substances the immune system will recognize. A variety of immune cell receptors are the result of acquired genetic knowledge of microbes. There's over 25,000 different genes that code for up to 1 billion different types of lymphocyte antigen receptors. There's a huge variety of receptors. The gene segments are shuffled around, resulting in many combinations. Lymphocyte education during maturation. So T cells mature in the thymus under negative and positive selection pressures. These are called tests. Positive selection process selects for T cells capable of recognizing self-MHC proteins. Those that can't are destroyed by apoptosis. Negative selection prompts apoptosis of T cells that bind to self-antigens displayed by self-MHC. And the process is called clonal deletion, which ensures self-tolerance. 0.9 T cell education in the thymus. So positive selection. In positive selection, T cells must recognize the self-major histocompatibility proteins. Failure to recognize self-MHC cells results in apoptosis, which is programmed cell death. Recognizing self-MHC results in survival. Survivors proceed to negative selection. So T cells must not recognize self-antigens. Recognizing self-antigens results in apoptosis. This eliminates self-reactive T cells that could cause autoimmune diseases. So failure to recognize self-antigen results in survival and continued maturation. Antigen-presenting cells. These engulf antigens and present the fragments of the antigens to T cells for recognition. There are 3 major types: dendritic cells, macrophages, and B cells. Dendritic cells are found in connective tissues and the epidermis. They act as mobile sentinels of boundary tissues. They phagocytize pathogens that enter the tissues, then enter the lymphatics to present antigens to T cells in lymph nodes. It's the most effective antigen presenter known, the key link between the innate and the adaptive immunity. Here's a figure of figure 2110. This is what a dendritic cell looks like. It's very branched. Macrophages are widely distributed in connective tissues and lymphoid organs. They present antigens to T cells, which not only activates the T cell, but also further activates the macrophage. An activated macrophage becomes a voracious phagocytic killer. They also trigger powerful inflammatory responses and recruit additional defenses. B lymphocytes do not activate naive T cells. They present the antigens to helper T cell to assist their own activation. Here in table 21.4 is an overview of B and T lymphocytes and a summary of the type of immune response, whether they secrete antibodies, their primary targets, the site of origin, site of maturation, the effector cells, and whether there's memory cell formation. Section 21.5, humoral immune response. So when B cell encounters a target antigen, it provokes a humoral immune response. So antibodies specific for that particular antigen are then produced. B cells, so activation and differentiation of B cells. B cells are activated when antigens bind to surface receptors, cross-linking them. And it triggers the receptor-mediated endocytosis of cross-linked antigen receptor complexes in clonal selection, which leads to proliferation and differentiation of B cells into effector cells. Most clone cells become plasma cells, which are antibody-secreting effector cells. These secrete specific antibodies at a rate of 2,000 molecules per second for 4 to 5 days, and then they die. Antibodies circulate in blood or lymph, binding to free antigens, marking them for destruction by the innate or other adaptive mechanisms. 2111, clonal selection of a B cell. So here the primary response is that initial encounter with the antigen. And only one of those specific lymphocytes is going to proliferate to form a clone. So the activated B cells are going to differentiate into plasma cells and memory B cells. And the plasma cells are the effector B cells because they are the ones that are responsible for secreting the antibody molecules. The memory B cell is primed to respond to the same antigen upon second exposure. Clone cells that do not become plasma cells become memory cells, which provide immunological memory and mount an immediate response to future exposures to the same antigen. Figure 2111, clonal selection of a B cell. So secondary response can be years later, months later, or years later. So a subsequent challenge by the same antigen results in a larger, more rapid response because of the memory cells. Let's talk about immunological memory. So primary immune response. In a primary immune response is cell proliferation and differentiation upon exposure to the antigen for the first time. So there's a lag period between 3 to 6 days, and then peak levels of plasma antibody are reached in 10 days. The antibody levels then will decline. Secondary immune response is when there is a re-exposure to the same antigen, and this gives a faster, more prolonged, more effective response. Sensitized memory cells provide immunological memory. They respond within hours, not days, and the antibody levels peak in 2 to 3 days at much higher levels. Antibodies bind with greater affinity, and the antibody level can remain high for weeks to months. 12, primary and secondary humoral responses. So here on the x-axis we have time in days, and on the y-axis we have the antibody titer, which is the antibody concentration in plasma in arbitrary units. So upon first exposure to an antigen A, there's a lag period before the antibodies are produced. So a primary immune response to the antigen A occurs after a delay. And then upon a second exposure to antigen A, first exposure to antigen B, so a secondary immune response to antigen A is faster and larger, as you notice from the graph in blue. A primary immune response to antigen B is similar to that for antigen A, because it's a different antigen, basically. So now we're going to talk about active and passive humoral immunity. So in active humoral immunity, this occurs when the B cells encounter antigens and produce the specific antibodies against them. There are two types of active humoral immunity. First one is through naturally acquired, which is formed in response to the actual bacteria or viral infection. In artificially acquired, it's formed in response to a vaccine of dead or attenuated pathogens. This will provide antigenic determinants that are immunogenic and reactive, and they also spare us the symptoms of a primary response. So that's the purpose of a vaccine. We don't want to get sick from the vaccine. We just want our immune system to be able to recognize whatever the antigenic determinant, to be able to mount a quick response upon exposure. In passive or humoral immunity, this occurs when a ready-made antibodies are introduced into the body. So B cells are not challenged by antigens. Immunological memory does not occur. Protection ends when the antibodies degrade. There are two types of passive humoral immunity. The first one is called naturally acquired. It's when the antibodies are delivered to the fetus via the placenta or to the infant through breastfeeding. And the second one is artificially acquired. It's the injection of serum, such as gamma globulin, and the protection is immediate but ends when the antibodies naturally degrade in the body. So figure 2113, active and passive humoral immunity. So humoral immunity, there's active and passive. So if we look at active, naturally acquired, this is when you get sick with a natural infection because you're in contact with that pathogen, and you develop immunity to it. Artificially acquired is through vaccination, where you get dead or attenuated pathogens, so changed in some way. In a passive immunity, naturally acquired is through the mother, right? From the mother to the fetus via the placenta. And this is naturally, so it's antibodies that are passed from the mother to the fetus, or from the mother through her milk to her infant. And artificially acquired instead is the injection of antibodies, and they were talking about this for COVID, of doing that. Like, so if somebody was sick, had been sick and recovered from COVID, they would take their antibodies and then give it to someone who is struggling with COVID. So on the subject of antibodies, these are also called immunoglobulins. They're proteins secreted by the plasma cells. They're made up of gamma globulin. It's the gamma globulin portion of blood. They're capable of binding specifically with antigen detected by the B cells. They're grouped into one of 5 Ig classes. So basic antibody structure. So overall, they have this T or Y-shaped. It's an antibody monomer, which consists of 4 looping polypeptide chains linked by disulfide bonds. The 4 chains consist of 2 identical heavy chains with a hinge region at the middles, and 2 identical light chains. The variable V regions on one end of each arm combine to form 2 identical antigen binding sites. The stems make up the constant regions. It's the area that determines the antibody class. It's considered the effector region of the antibody. Serves a common function in all antibodies, dictating 1, the type of cell and or chemicals that the antibody can bind to, and 2, how the antibody class functions to eliminate antigens. For example, some antibodies fix complement, some circulate in blood, and some are found in body secretions. Figure 2114a, antibody structure. So here we can see the heavy chain, the light chain, and that's the antigen binding site. And then we have the stem region. And notice the disulfide bonds keeping those polypeptide chains together. There are 5 major classes of antibodies: IgM, IgA, IgD, IgG, and IgE. So IgM is a pentamer. They're the largest ones. It's the first antibody that is released. They have it's a potent agglutinating agent and readily fixes and activates complement. Second one is IgA. It's a monomer. It could be a monomer or a dimer. It's found in mucus and other secretions, and it helps prevent entry of pathogens because it's found in strategic areas of the body, like near the nose, near the mouth. Table 21.5, immunoglobulin classes. So here's the IgM. Notice it's a large pentamer, and the IgA is shown here as a dimer. Then there's IgD, which is a monomer that is attached to the surface of B cells, and it functions as a B cell receptor. IgG is a monomer. 75 to 85% of antibodies in plasma are IgGs. They form the secondary and late primary responses, and it crosses the placental barrier. IgE is a monomer that's active in some allergies and parasitic infections, and it causes mast cells and basophils to release histamine. In this figure, we can see the IgD, the IgG, and IgE. Plasma B cells can switch from making one class of antibodies to another. So they retain specificity for the same antigen. The IgM is released during the primary response, but plasma cell can switch to IgG for a secondary response. almost all secondary responses are IgG. Switching from IgM to IgA or Ig can also occur. Antibody targets and functions. Antibodies do not destroy antigens, they simply inactivate and tag them. They form antigen-antibody immune complexes. Defensive mechanisms used by antibodies are neutralization, agglutination, precipitation, and complement fixation. Neutralization is the simplest but one of the most important defensive mechanisms. Antibodies block a specific site on viruses or bacterial exotoxins. It prevents the antigens from binding to receptors on tissue cells. Antigen-antibody complexes undergo phagocytosis. Agglutination is when antibodies that can bind the same determinant on two different antigens at the same time. Each antibody has two arms, each containing a variable region capable of binding to one antigen. This allows for antigen-antibody complexes to become cross-linked into a large lattice-like clump. The process is referred to as agglutination, and a very good example of this is seen in the clumping of mismatched blood cells. Precipitation, soluble molecules instead of cells are cross-linked into complexes, and the complexes precipitate out of a solution. The precipitated complexes are easier for phagocytes to engulf. Complement fixation and activation. So the main antibody defense against cellular antigens, like bacteria and mismatched red blood cells. When several antibodies are bound close together on the same antigen, complement binding sites on their stem regions are aligned. Alignment triggers complement fixation, which leads to cell lysis as well as other complement functions. For example, amplification of the inflammatory response and opsonization to help with phagocytosis. So this figure shows the effects of antigen-antibody binding. So it will trigger the complement activation, and which leads to cell lysis. Remember, when complement is activated, the cell will lyse because it will, through a cascade reaction, it will cause the insertion of those holes into the cell, right? And so things will leak out and things will go in. Also, it inactivates by neutralization masks of dangerous parts of bacterial exotoxins and viruses. Okay, so here, if you notice, the antibody surrounds a virus. In agglutination, seen like with the mismatched red blood cells, so cell-bound antigens, and precipitation, where these the antibodies will bind to soluble antigens and they will precipitate. And agglutination, neutralization, and precipitation will enhance phagocytosis, and precipitation and complement activation will enhance inflammation. Clinical homeostatic imbalance 21.2. Parasitic infections by worms such as Ascaris and Schistosoma require different immune attack strategies. Worms are too big for regular plan attack, in other words, precipitation, lysis, agglutination, or neutralization. So IgE antibodies still play a critical role in worms' destruction by binding to the surface of the worm, marking it for destruction by eosinophils. Eosinophils then bind to the exposed stems of IgE, which triggers the eosinophils to release their toxic contents onto the prey, lysing it from the outside. Clonal antibodies are can be used as a clinical and research tool. So what are monoclonal antibodies? They're commercially prepared pure antibodies that are specific for a single antigenic determinant. And that's what they were finding with COVID-19. So giving a patient antibodies directly from a patient wasn't working very well. So they came, they thought that monoclonal antibodies had a better specificity, basically. So how do you prepare them? How do you make these types of antibodies? They're produced by hybridomas, which are cell hybrids that are formed from the fusion of a tumor cell and a B cell. So the tumor cell has that ability to proliferate indefinitely, while the B cell portion allows for the production of a single type of antibody, making it very specific. So it can be used in research, clinical testing, cancer treatment, and also, like I said before, for COVID-19. Summary of antibody action. So antigen-antibody complexes do not destroy the antigens, they only prepare them for destruction by the innate defenses. Antibodies go after extracellular pathogens, they do not invade solid tissue unless a lesion is present. Recent exceptions found, antibodies can act intracellularly if attached to a virus before it enters a cell. And it activates mechanisms that will destroy a virus. Section 21.6, cellular immune response. So T cells provide defense against intracellular antigens. An example is the cells infected with viruses or bacteria, cancerous or abnormal cells, or foreign transplanted cells. Some T cells directly kill cells, others will release chemicals that regulate the immune response. T cells are more complex than B cells, both in classification and function. Two populations of T cells are based on which cell differentiation glycoprotein receptors are displayed on their surface. We have CD4 cells, which usually become helper T cells that can activate B cells, other T cells, and macrophages. This is a direct adaptive immune response. Some will become regulatory T cells, which moderate the immune response. The other can also become memory T cells. CD8 cells, these become cytotoxic T cells that are capable of destroying cells harboring foreign antigens. And these can also become memory T cells. Helper, cytotoxic, and regulatory T cells are activated T cells. Naive T cells are simply termed CD4 or CD8 cells. Figure 21.16, major types of T cells. So we have an immature lymphocyte that can, from the red bone marrow, that can differentiate into a CD4 cell or a CD8 cell. CD4 cell, once it's activated, and it's going to, activation is going to include its binding to a class 2 MHC protein displaying an antigen, so something from a dendritic cell, and then it's going to proliferate and become either helper T cells or regulatory cells. There will be some component that is going to turn into memory cells. Whereas a CD8 cell, once it's activated by, again, like an antigen-presenting cell, like a dendritic cell, and this one here is by a class 1 MHC protein displaying antigen, this is going to differentiate into cytotoxic T cells. MHC proteins and antigen-presenting cells. So T cells respond only to processed fragments of antigens displayed on the surfaces of cells by the major histocompatibility complex proteins. Antigen presentation is vital for activation of naive T cells and the normal functioning of effector T cells. So in fact, there are two classes of MHC proteins. The class 1 MHC proteins is displayed by all cells except red blood cells. And class 2 MHC proteins, these are only displayed by antigen-presenting cells, for example, dendritic cells, macrophages, and B cells. And both types are synthesized in the endoplasmic reticulum and bind to peptide fragments. So class 1 MHC proteins bind with short fragment, about 8 to 9 amino acids of endogenous antigen, protein synthesized inside a cell. Endogenous antigen can be self-antigen, in other words, normal proteins of the cell, or non-self-antigen, which are abnormal proteins found in infected or abnormal cells. The class 1 MHC is crucial for CD8 cell activation. It acts as antigen holders that form self-part that the T cells will recognize. They also inform cytotoxic T cells of microorganisms hiding in cells. Cytotoxic T cells ignore displayed self-antigens. And the class 2 MHC proteins, these bind with longer fragments, about 14 to 17 amino acids, of exogenous or extracellular antigens that have been engulfed and broken down in a phagal lysosome by an antigen-presenting cell. The class 2 MHC proteins are recognized by helper T cells, and they signal the CD4 cells that help is required. Table 21.6, the role of MHC proteins in cellular immunity. So here we see the two types, the class 1 on the left and class 2 on the right. And like for example, class 1 is displayed by all nucleated cells, in other words, not red blood cells because they're not nucleated. And class 2 are displayed only by antigen-presenting cells like dendritic cells, macrophages, and B cells. And then it shows class 1 is recognized by CD8 cells and cytotoxic T cells, whereas class 2 proteins are recognized by naive CD4 cells and helper T cells. And then what do they react to? And cells displaying foreign antigen on MHC send what message? MHC restriction. So CD4 and CD8 cells have different requirements for MHC protein that presents antigens to them. The CD4 cells that become T helper cells are restricted to binding to only class 2 MHC proteins, typically on antigen-presenting surfaces, whereas the CD8 cells that become cytotoxic T cells are restricted to binding only class 1 MHC proteins found on every cell surface, including APC surfaces. Once activated, the cytotoxic T cells seek out the same antigen on the class 1 MHC proteins on any cell. Specific effector T cells. So helper T cells play a central role in the adaptive immune response. They activate both humoral and cellular arms. Once primed by the antigen-presenting cell presentation of the antigen, helper T cells, it helps activate the B cells and other T cells. It induces the T and B cell proliferation and secretes cytokines that recruit other immune cells. Without the T helper cells, there's no immune response. Activation of B cells. Helper T cells interact directly with B cells, displaying antigen fragments bound to MHC2 receptors. This also stimulates B cells to divide more rapidly and begin antibody formation. And B cells may be activated without T helper cells by binding to T cell independent antigens. And the response is weak and short-lived. But most antigens are T cell dependent antigens that require T helper cell co-stimulation to activate B cells. (2118a), the central role of helper T cells in mobilizing both humoral and cellular immunity. So here we can see a helper T cell that helps in humoral immunity. So there's the helper T cell, the T cell receptor, and the MHC protein of the B cell displaying a processed antigen. So here is a B cell that is being activated. So in one, the T helper cell binds with the non-self complexes of a B cell that has encountered its antigen and is displaying it on the MHC2 on a surface. And that the T helper cell is going to release interleukins as co-stimulatory signals to complete B cell activation. Activation of CD8 cells. CD8 cells require T helper cells to become activated into destructive cytotoxic T cells, and this caused dendritic cells to express co-stimulatory molecules required for CD8 cell activation. So figure 2118, the central role of helper T cells in mobilizing both humoral and cellular immunity. So in step 1, so what do we see here? We see an antigen-presenting cell. They're showing a dendritic cell, a helper T cell, and a CD8 T cell. So the T helper cell binds the dendritic cell. The T helper cell stimulates the dendritic cell to express co-stimulatory molecules. And in step 3, the dendritic cell can now activate the CD8 cell with the help of interleukin-2 secreted by the T helper cell. Cytotoxic T cells directly attack and kill other cells. The activated cytotoxic cells circulate in the blood and lymph and lymphoid organs in search of body cells displaying the antigen they recognize. An activated T cell target would be a virus-infected cell, cells with intracellular bacteria or parasites, cancer cells, and foreign cells from transfusion or transplants. Cytotoxic T cells deliver the lethal hit using 2 mechanisms. The first one, these cells release perforins and granzymes by exocytosis. Perforins create pores through which granzymes enter the target cell, and granzymes will stimulate apoptosis. So the cell is just going to die. A second mechanism is the T cell binds specific membrane receptor on the target cell and stimulates apoptosis. Figure 2119a, cytotoxic T cells attack infected and cancerous cells. So the cytotoxic T cell identifies foreign antigens on the MHC1 proteins and binds tightly to the target cell. And then it releases perforin and granzymes, these molecules, from its granules by exocytosis. Perforin molecules insert into the target cell membrane, polymerize, and form transmembrane pores, similar to those produced by complement activation. The granzymes enter the target cell via the pores. Once inside, the granzymes activate enzymes that trigger apoptosis. And in 5, the TC detaches and searches for another prey. Figure 2119b shows a cytotoxic T cell that attacks an infected and cancerous cell. Cytotoxic T cells, so natural killer cells recognize other signs of abnormality that cytotoxic T cells do not look for, such as cells that lack class 1 MHC proteins, antibodies coating target cell, and different surface markers seen on stress cells. Natural killer cells use the same key mechanisms such as the TC cells for killing their target cells. Immune surveillance is when natural killer and cytotoxic T cells prowl the body looking for markers they each recognize. 0.8 cells and molecules of the adaptive immune response. So this table summarizes the cells and their function in the immune response. It talks about the B cells, the plasma cells, T cells, so helper cells, cytotoxic T cells. Memory, we didn't really talk about regulatory T cells. There's memory cells, antigen-presenting cells like a dendritic cell, and then different types of molecules like antigens, an antibody, perforins and granzymes, complement, and cytokines, which I kind of skipped over. So that's it for this year. Quite a semester, but we made it through it. Good luck.
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then, the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the cytotoxic T cell is activated and clonal expansion occurs, resulting in some long-lasting memory cells ready for a subsequent exposure, and many active cytotoxic T cells, or CTLs, that leave the patient's lymph vessel to search for infected cells that present rhinovirus epitope with their MHC1. The epitope is recognized by the TCR of the cytotoxic T cell, and the MHC1 is recognized by the T cell's CD8 glycoprotein. The cytotoxic T cell then secretes perforin molecules, which form channels in the infected cell's membrane. Granzyme, also from the T cell, passes through the perforin channels and activates enzymes that lead to apoptosis, the programmed death of the infected cell. The cytotoxic T cell then moves on to look for another infected cell. Together, the humoral and cellular defenses are the body's third line of defense in fighting a very specific pathogen. The humoral defenses, ultimately in the form of antibodies, conduct surveillance for rhinoviruses that are outside the body cells, in the patient's tissue spaces and bloodstream. The cellular defenses, in the form of cytotoxic T cells, search for rhinovirus-infected cells, inducing apoptosis when they are found. Memory T and B cells help our patient's body to recognize and more rapidly fight the same rhinovirus should it infect her again.